Hi viewers, today we are going to discuss the acquired immunity apart from the types of immunity. As we know, the immunity get classified into two major types, innate and the acquired immunity. Now the acquired immunity can be defined as the resistance that an individual acquires during lifetime by recognizing and selectively eliminating specific foreign molecules is known as the acquired immunity. That is, acquired immunity is a more specialized form of immunity. It developed late in evolution and is found only in vertebrates. There are certain characteristic features of the acquired immunity. There are four main characteristic features, namely antigenic specificity, diversity, immunological memory in the form of primary response and secondary response, and fourth one is the self and non-self recognition. Let us see one by one. Antigenic specificity. The immune system or antibodies can distinguish among antigens even between two proteins that differ in only one amino acid. Such a keen specificity is shown by the acquired immune system. And second one is the diversity. The immune system is capable of generating enormous antibody diversity in its recognition molecules, allowing it to recognize billions of unique structures or patterns on foreign antigens, genes from the basis of such diversity. Coming to the immunologic memory, the immune system exhibits memory on the second encounter of the same antigen by generating a secondary response which is more specific, high-end and quick. What is the primary response is, when the individual is exposed to an antigen for the first time, the production of antibodies takes place. The number of antibodies are less and the time taken for the production of antibodies is more. The immune system memorizes the structure and pattern of the antigens. Whereas the second response is, for the second time, if similar antigen entered into the body, more antibodies are produced in short span of time. This is due to the subsequent exposure to that particular antigen which has attacked previously. That is, primary response, it is a slow response, whereas the second response is a exaggerated response. And the fourth one is the self and non-self recognition. Self-tolerance is one of the unique characteristics of the immune system which prevents it from reacting to the body's own molecules while still effectively eliminating foreign antigens. Multiple mechanisms ensure self-tolerance. Failure of these mechanisms may lead to autoimmunity. So, antigenic specificity, diversity, immunologic memory and self and non-self recognition are the main characteristic features of the acquired immunity. And here we will see types of acquired immunity. The acquired immunity is divided into active immunity, active acquired immunity and the passive acquired immunity. Acquired immunity is classified into two types, active acquired immunity and the passive acquired immunity. Where in the active acquired immunity, the individual's immune system plays active role in providing the immunity, whereas in the passive acquired immunity, the host or individual's immune system may not play an important role, it keeps quiet, okay, and the antibodies which are injected into the body of the individual, they may take care of the providing immunity. And again the active immunity again divided into two types, natural active acquired immunity and artificial active acquired immunity. In the same way, passive immunity is also get classified into two types. Now, natural passive acquired immunity and artificial passive acquired immunity. So both the active and passive immunities get classified into natural one and the artificial one. See the active acquired immunity. This is the resistance developed by an individual as a result of an antigenic stimulus. It is also known as the adaptive immunity, as it represents the adaptive response of the host to a specific pathogen or other antigens. It involves the active functioning of the host's immune apparatus, leading to the synthesis of antibodies and the production of immunologically active cells. It sets in only after a latent period which is required for the immunological machinery to be set in motion. During the development of active immunity, there is often a negative phase during which the level of measurable immunity may actually be lower than it was before the antigen stimulus. This is because the antigen combines with any pre-existing antibody and lowers its level in circulation. Once developed, active immunity is long lasting. If an individual who has been actively immunized against an antigen experiences the same antigen subsequently, the immune response occurs more quickly and abundantly than during the first encounter. Besides the development of humoral and cellular immunity, active immunity is associated with immunologic memory. Active immunization is more effective and confers better protection than the passive immunity. And come to the different kinds of active immunity, that is natural active immunity, active acquired immunity, and artificial active acquired immunity. First one is the natural active acquired immunity. This natural active acquired immunity, it is results either a clinical or an inapparent infection by a microbe. A person who has recovered from an attack of measles develop natural active immunity. The large majority of adults in the developing countries possess natural active immunity to poliomyelitis due to repeated inapparent infections in childhood. Such immunity is usually long lasting, but the duration varies with the type of pathogen. The immunity is lifelong, following many viral diseases such as chickenpox and the measles. The immunity appears to be short-lived, such as influenza or common cold. Influenza can recur in an individual after a few months or a year, but this is not so much due to lack of the immunizing capacity of the virus as to its ability to undergo antigenic variation, so that immunity following the first infection is not effective against the second infection caused by an antigenically novel virus. In the common cold, the apparent lack of immunity is because the same clinical picture can be caused by infection with a large number of different viruses. The immunity following bacterial infection is generally less permanent than that of following viral infection. This is about the natural active acquired immunity. And coming to the artificial active acquired immunity, it is the resistance induced by vaccines. Vaccines are preparations of live or killed microorganisms or their products used for immunization. The best examples for artificial active acquired immunity are the vaccines. So this is, here we are seeing the COVID-19 vaccine. These vaccines are different types. There are bacterial vaccines, viral vaccines. Out of the bacterial viral, we may use live organisms for the preparation of vaccines or killed organisms for the preparation of vaccines. See the bacterial vaccines, the best examples are live BCG vaccine for tuberculosis, BCG means Bacillus calmette guerin. BCG vaccine, it is a live vaccine. And for the killed vaccine, the best example is the cholera vaccine. The subunit vaccine is the typhoid antigen vaccine. And some of the times bacterial products can be used as the vaccines like the TT, tetanus toxoid. Tetanus toxoid. These all are the bacterial vaccines. Coming to the viral vaccines, apart from the viral vaccines, live oral polio vaccine, which is called as the Sabin vaccine, and killed polio vaccine, which is intramuscularly injectable, that is SALK vaccine, and subunit vaccine, the best example is the hepatitis B vaccine. See, this is the polio drops, oral polio drops, and this is the injectable polio vaccine. And what is the Sabin SALK are the names of the scientists? Albert Sabin, who prepared the oral polio drops, and Jonas Salk, who prepared the injectable polio vaccine. And there are live vaccines which are prepared from the living microorganisms, and there are killed vaccines prepared from the killed microorganisms. What are the live vaccines? They initiate infection. Live vaccines, they initiate infection without causing any injury or disease. The immunity following live vaccine administration therefore parallels that following natural infection, though it may be of a lower order. The immunity lasts for several years, but booster doses may be necessary for the live vaccines. Whereas killed vaccines, they are less immunogenic than live vaccines, and protection lasts only for a short period. Therefore, they have to be administered repeatedly. Generally, at least 2 doses being required for the production of immunity. The first dose is the primary dose, and the subsequent dose is the booster dose. This is about the artificial active acquired immunity. And coming to the passive acquired immunity. This is the resistance that is transmitted in a ready-made form. Here the recipient's immune system plays no active role at all. There is no antigenic stimulus. Instead of that, pre-formed antibodies are directly administered. There is no latent period, protection being effective immediately after passive immunization. There is no negative phase. The immunity is transient, usually lasting for days or weeks, only till the passively transmitted antibodies are metabolized and eliminated. No secondary type response occur in passive immunity. In fact, passive immunity diminishes in effect with repetition. When a foreign antibody is administered a second time, it is eliminated more rapidly than initially. Following the first injection of an antibody such as immune horse serum, the elimination is only by metabolic breakdown, but during subsequent injections of horse serum, elimination is much quicker as it combines with antibodies to horse serum that would have been produced following its initial injection. This factor of immune elimination limits the usefulness of repeated passive immunization. Passive immunization is less effective than active immunization. The main advantage of passive immunization is that it acts immediately and therefore can be employed when instant immunity is desired. This passive acquired immunity is also classified into 2 types: natural passive acquired immunity, artificial passive acquired immunity. Natural passive acquired immunity, it is the resistance passively transferred from mother to baby. In human infants, maternal antibodies are transmitted predominantly through the placenta, while in animals such as pigs, transfer of antibodies occurs mainly orally through the colostrum, the first milk of the mother. The human colostrum, which is also rich in immunoglobulin A antibodies, IgA antibodies, resists to intestinal digestion, gives protection to the neonate. The human fetus acquires some ability to synthesize antibodies IgM from about the 20th week of life, but its immunological capacity is still inadequate at birth. It is only by about the age of 3 months, that the infant acquires some measure of immunological independence. Until then, maternal antibodies give passive protection against infectious diseases to the infant. It is for this reason the most pediatric infections are more common after the age of 3 months than in younger infants. By active immunization of mother during pregnancy, it is possible to improve the quality of passive immunity in infants. Immunization of pregnant women with tetanus toxoid is recommended for this purpose in communities in which neonatal tetanus is common. Coming to the artificial passive acquired immunity, it is the resistance passively transferred to a recipient by the administration of antibodies. The agents used for this purpose are hyperimmune serum of animal or human origin, convalescent serum, and pooled human gamma globulin, otherwise the pooled serum. These are used for prophylaxis or for the therapy. Let us see one by one the hyperimmune serum. Equine hyperimmune sera such as anti-tetanus serum ATS prepared from hyperimmunized horses used to be extensively employed. They give temporary protection but carried the disadvantages of hypersensitivity and immune elimination. Human hyperimmune globulin, the best examples are the tetanus immune globulin TIG, is free from those complications and also provides more lasting protection. Anti-sera of animal origin are now recommended only where human preparations are not available. The best examples for these are antivenom, anti-gangrene sera, and anti-botulinum sera. And coming to the convalescent serum, sera of patients recovering from infectious diseases contain high level of specific antibodies. Such kind of serum collected from patients who recovered, that is known as the convalescent serum. So nowadays the vaccines for measles and rubella are prepared in this method. So recently we have observed that the corona treated with this technique prior to the discovery of the vaccines. So they have collected the convalescent serum from the COVID-19 recovered patients, and that will be administered to the COVID-19 patients. Coming to the pooled serum. Pooled serum collected from the different healthy individuals and pooled, otherwise summed up, it contains antibodies against all common pathogens prevalent in the region. Human gamma globulin used for the passive immunization against some viral infections like hepatitis A. Human gamma globulin is also used in the treatment of patients with some immunodeficiencies. Passive immunization is indicated for immediate and temporary protection in a non-immune host faced with the threat of infection. Passive immunization may also be used for the suppression of active immunity when the active immunity may be injurious. The best example for this is the use of RH immunoglobulins during the delivery to prevent immune response to the RH factor, that is rhesus factor, in RH negative mother when there is a RH positive baby. This is about the pooled serum. And coming to the Combined immunity. Sometimes a combination of the active immunity methods and passive immunity methods of immunization are combinedly used. Ideally, whenever passive immunization is employed for immediate protection, combined immunization is to be preferred, as in the protection of a non-immune individual with a tetanus-prone wound. The method is to inject tetanus immunoglobulin TAG in one arm and the first dose of tetanus toxoid in the other arm. This is followed by the full course of phased tetanus toxoid injections. Tetanus immunoglobulin TAG provides the protection necessary till active immunity is able to take effect. Combined immunity. And the last one is adoptive immunity. A special type of immunization is the injection of immunologically competent lymphocytes. This is known as adoptive immunity and does not have general application. Instead of whole lymphocytes, an extract of immunologically competent lymphocytes known as the transfer factor can be used. This has been attempted in the treatment of certain types of diseases. The best example is the lepromatous leprosy. This is about the adoptive immunity. So, finally, the adaptive immunity is acquired immunity is the immunity that develops during the lifetime of the individual, classified into active immunity and passive immunity. In the active immunity, it develops in response to an infection or vaccination, whereas the passive immunity develops after you receive antibodies from someone or somewhere else. So, the best example for the natural active acquired immunity is antibodies developed in response to an infection, whereas artificial active acquired immunity, antibodies developed in response to vaccination. Natural passive acquired immunity, antibodies received from mother, example through the breast milk, and artificial passive acquired immunity, antibodies received from a medicine, example from the gamma globulin injection or from the infusion. So, this is all about the acquired immunity. By this, we will sum up the topic. Thank you, one and all.